Hey guys, and welcome to AP Help 101. So, last week I did begin talking about Heredity, but since Heredity is such a big unit, I decided to split it up into two PowerPoints. So, this week I'm going to talk more about Heredity. So, Heredity Part 2. So, just a few vocabulary words that we may need to know. Polygenic means traits that are a blending of several separate genes. Multifactorial basis is where there's an underlying genetic component with a significant environmental influence. So again, that just means that genetics do play a role, but environment also plays a role as well. And finally, we have nurture and nature, which is the development of intelligence is the result of an interaction of genetics and environment. And this is still up to debate because we're still trying to figure out if environment plays a big role, or if genetics plays a big role, or maybe if they both combined together play a big role. So today we're going to start talking about linked genes, sex-linked genes, mutations, and things like that. So linked genes are genes that are on the same chromosome, and they tend to be inherited together unless they are separated during crossover. Remember that crossover is what happens during meiosis when the genes switch on both chromosomes. Now we have 44 autosomes and 2 sex chromosomes, so 46 chromosomes together, 23 pairs. So one of the types of linkages is called sex linkage, and sex-linked traits are traits that are located on the X chromosome. Now, why the X chromosome? Well, it's because the Y chromosome isn't necessarily very large. It's very small. It doesn't really contain a lot of genes on it. And so most of the genes that are sex-linked are located on the X chromosome. Remember, women have 2 X chromosomes, whereas men have an X and a Y chromosome. So if there is a recessive disease that is sex-linked, which means it'll be located on the X chromosome, women will only have that recessive mutation if both X chromosomes carry the recessive allele. And for that to happen, both the mother and the father have to carry the disease. The father actually must be exhibiting that mutation, but the mother just needs to be a carrier for a woman to have the disease. Now, if girls only have one chromosome with the mutation, they will not express that trait or that disease, but they will be carriers of that disease, which means their children may or may not get the disease. Men, on the other hand, since they only have one X, they will receive that mutation if that X has that mutation. That Y chromosome won't cancel out that X. Now, with a dominant mutation, it's a little different because the X chromosome is so-called dominant in this case. So with just one mutated X chromosome, both men and women will exhibit that trait or that disease. So being with the dominant mutations are very rare. They're not usually significant. They're not significantly large in a way. So really, dominant mutations are rare, but when they do happen, men and women are equally affected. So an example of a recessive sex-linked trait would be color blindness and hemophilia. Now, if you do some research on color blindness and hemophilia, you'll notice that most of the people who have these diseases are men, because men are more likely to carry that one mutated X chromosome. So just a little bit on crossover and linkage mapping. So remember, crossover is when the genes are switched in between chromosomes. So the further apart two genes are on a chromosome, the more likely they are to be separated during crossover. So if one gene is on one end of the chromosome and the other gene's on the other end of the chromosome, they are likely to be separated, but if they're right next to each other on the chromosome, they most likely will stay together. So where crossover and recombination occurs, there you may see what is called a chiasma, which is just a physical bridge built around that point. And just another vocab word to remember, a linkage map basically shows the rate of crossover from the greatest to the least or least to greatest. So say there are 3 genes. We have gene B, A, and D. And the rates for them are, between B and A is 5%, between A and D is 10%, and between B and D is 25%. If you want to make a linkage map, you put B, A, and D in that order because it shows from least to greatest, least to greatest. In that order, B, A, D, the least to the greatest percentage of being crossover. A pedigree. A pedigree is a family tree which shows the phenotype of one trait being studied for every member of the family. And pedigrees play a huge role, especially in the AP Bio test, but they're really not that difficult to read once you get a hang of it. So these are used to determine how a certain trait is inherited. And these pedigrees can go back multiple generations. They can go back 2, 3 generations. Females are usually depicted by a circle, males are depicted by a square. Now, if that circle or square is fully shaded, that person does exhibit that trait. And if the circle or square is half shaded, then that person is a carrier. And if it's not shaded, then they aren't affected at all. So this is a pedigree. As you can see, the squares are the males and the circles are the females. At the top, you'll notice we have the first generation, and below that, the second generation, which is their children. And below that, we have the third generation, which is those children's children. So the shaded ones are the ones that are affected, and the ones that are not shaded, which means they are not affected. They don't show the carriers in this case, but if someone were to be a carrier, only half of the circle or half of the square would be shaded. The lines crossed through them means that they are deceased, so looks like only the last generation exists in this case. Everyone else is deceased. But these can be great for finding out how a trait is inherited. Looks like this may be sex-linked because mainly the males are affected by this and not so much the females. So now we have X inactivation. And this is an interesting phenomenon actually. Early in development, one of the X chromosomes in a woman is inactivated at random, which is odd, you may think. You're really only expressing one of your X chromosomes. So all of the cells of female mammals are actually not the same. This X inactivation creates a dark black spot at the outer edge of the nucleus, which is called a Barr body. This is really interesting because, again, you would think that you're expressing both chromosomes, but you're actually not. There's just one chromosome that's being expressed, and the other one just randomly was inactivated. Mutations. Mutations are changes in the genome, and we have two types of mutations, gene mutations and chromosomal mutations. Gene mutations affect the gene itself, and chromosomal mutations affect the chromosome. So some examples of genetic disorders, which are gene mutation related, are cystic fibrosis, Huntington's disease, color blindness. Cystic fibrosis is autosomal recessive, so it's based off a recessive disease trait. Huntington's is autosomal dominant, so the dominant allele is affected. And color blindness, again, is sex-linked recessive. Some examples of chromosomal disorders are Down syndrome, as well as Turner syndrome. Chromosomal aberrations. These can be seen using a karyotype. Unlike genetic aberrations, we can't really see any changes in the gene yet. Hopefully we'll get there. But we can see chromosomal changes. Karyotypes show the size, number, and shape of chromosomes, and they can show chromosomal abnormalities. So you can see if we're missing a part of a chromosome, or if there are more chromosomes than necessary, or if there are less chromosomes than necessary. So these are 4 different types of aberrations. We have deletion, which is a fragment lacking a centromere, is lost during cell division. Inversion is when a chromosomal fragment reattaches to its original chromosome, but in a reverse orientation. Translocation is when a part of a chromosome becomes attached to non-homologous chromosomes. And polyploidy is when we have an extra set of chromosomes. And finally, we have non-disjunction, and that is when homologous chromosomes fail to separate. So a generalized term for that is aneuploidy, which is any abnormal number of chromosomes. A more specific term for that would be trisomy, which is where you have 3 chromosomes instead of 2. And that is important to remember because, again, trisomy 21, which results in Down syndrome. And you don't need to necessarily know all of the diseases and what causes them all, but it is important to remember Down syndrome, especially because it's one of the more common diseases than the other ones. So I hope you enjoyed this video. If you have any questions or comments, leave them down below, and I will see you guys next week.